Okay, so we're still in meiosis 1. We've just finished up prophase 1 where those homologous chromosomes got together and crossed over. So now we're entering metaphase 1, the second phase of meiosis 1. And this is the shortest phase. Tetrads, remember those two homologous chromosomes, line up at the equator. Crossing over has already occurred, so we could have some weird combinations. But what I want to show you about metaphase is that this is where independent assortment occurs. And when I say independent assortment, I mean that tetrad with your maternal and paternal chromosomes can line up in either direction along the equator, along that metaphase plate. And so here's the first possibility, which is where you have mom's genes on the left and dad's genes on the right, and they are all just lined up very nicely. Another possibility is that maybe that tetrad inverted. And so you wind up with one parent's chromosome on the right and one parent's chromosome on the left, and then you have it flipped further down the line. And this might, seem fairly, trivial with an organism that only has a few chromosomes, but you have 23 pairs of chromosomes. And so when you're making gametes, you can wind up with numerous combinations or numerous permutations of chromosome orientation, where you can make all sorts of different unique individuals, if you will, different kinds of gametes, just because of this process of independent assortment. So this is our second mode. This is our second way of increasing genetic variation. The first was recombination. The second here is independent assortment. So in metaphase 1, your homologous chromosomes, your tetrads, line up along the metaphase plate, and independent assortment can occur. So here we have this one that's crossed over, and these chromosomes can line up in either direction. There's nothing preventing these chromosomes from lining up in any orientation. The next phase is anaphase 1, and this basically the tetrad separates, or the homologous chromosome separates, and they move to different poles. So there goes. I'll show this again, because this is just overviewing the entire process. So here we have prophase 1, crossing over occurs. Metaphase 1, they're actually separating those to the poles. So you're getting sort of one homolog in each pole, and it's going to separate. But let's keep going through. So anaphase 1, your homologs separate. The final phase of metaphase 1 is telophase 1 / cytokinesis. And each pole now has one set of chromosomes, and they're going to actually get separated and split into two. So here's your cytokinesis. Your cleavage furrow is forming between these two, and what you wind up at the end is two haploid cells, each of which contains unique chromosomes. So you see how this haploid cell is very different from this haploid cell. In the next lecture, we're going to discuss meiosis 2, where those sister chromatids are actually going to separate.